So first of all, welcome to the third talk in the conversation series. And this is a institute holiday, and despite that, you turned up. So thanks again once for that. So our speaker today is Dr. Chitra Pattabiraman, who's a consultant in pathogen genomics. So she's trained as a virologist and molecular biologist who uses genomic tools, for example, sequencing, to identify pathogens. She obtained her integrated PhD in life sciences from National Center for Biological Sciences, NCBS, in Bangalore. So she has been awarded the SCRP Royal Society Newton International Fellowship and the Indian Alliance Early Career Fellowship for postdoctoral work on brain infections. In fact, she will be giving a technical talk, a biology seminar on Monday afternoon on finding the pathogens in the brain and blood. But today she'll be talking about her work on which actually has been very effective during the COVID pandemic. So she's consults with multiple organizations on projects related to infectious diseases, emerging infections, and public health in India. So without further ado, let me invite Dr. Pattabiraman to give a talk on ways of the virus. So it's going to be a 30-minute talk, and there will be a moderated session of questions and answers from the audience following that.
Thank you. Hi, good afternoon, everyone. So I'm Chitra, and thank you for inviting me. It's really kind. And this is a public talk, so I thought the idea is to give you a flavor for the ways of the virus. This is broadly going to be about how we use genomics to track how viruses emerge, go all over the world, and sometimes disappear. But the way I've formatted this talk is that we have about 15 minutes of actual talk, and then we are going to play a game. So all of you are going to participate in a video game. And the hope is that the information that I convey to you in the first 15 minutes will help us to win the game. So we'll see. So human pathogens are as old as humans themselves. And this is a person with polio, and this is a person these are people infected with smallpox. Now, smallpox is a virus that actually got transported to South America from Europe, and the people there had no immunity to it, and they actually succumbed in really large numbers. So this is a Florentine Codex, since all of you have been in a workshop for the last week on inscriptions. So this is the Spanish flu. This was a slide I used to use before the pandemic to talk about how bad a respiratory virus can be. This is no longer required. It's a small world for a virus, and it has been this way for a while. Viruses can get around. So these are, I think, a few of the airports in the world are connected, or the first image is of air traffic around the world. And I think it's only for some of the airports. It's not all of the possible routes. And the second image is of shipping routes. So you can see that most places on the planet are really connected to each other. And what we know about infections, particularly of certain type called zoonotic infections that humans get from wildlife, we have some idea of the kind of risks that different regions of the world have for these kind of events. And these are based on things like population density, wildlife diversity, the contact between human beings and wildlife, and so on. So the spots that you see in red here, that's what it is based on. And you can see there are many regions of the world, both parts of Europe, Southeast Asia, as well as parts of South America and Central America. Are hotspots, or these are predicted hotspots. So diseases can emerge anywhere. And this is the idea that we have of how a pathogen might come into human beings, right? So what you see in the background here is basically this wave that you see in the background is basically of animals getting infected in the wild. then recovering at some rate, or the virus going down, then some susceptible animals become available again. So there's a natural pattern of that particular virus. Let's assume it's a virus in this case. And every once in a while, this virus then comes into contact with domestic animals, vectors, or other amplifying hosts. And when this happens, the virus then has to adapt. So this is for a successful spillover to happen, you would want this virus to adapt to a new host and amplify, preferably amplify in that host, so make a lot of copies of itself. So enough of that virus is available for a second spillover event into human beings, right? And this is why we think that where there is a contact between wildlife and human beings, or like where there's a contact with domestic animals and human beings, and as you increase the number of such contacts, you may increase the likelihood of spillover events. We believe that most of these spillovers don't actually give rise to anything to worry about. However, once in a while, there might be a virus that acquires the properties to amplify in human beings as well. When that happens, you get amplification in human beings. Again, this is likely going to be a local outbreak. So whoever is in contact. So we know that for infectious diseases, once human beings started congregating and living in settlements, that's when we start seeing outbreaks as well. So and so basically this would then die down. However, now if these human beings are mobile, or if there are ways to connect these people to another place, you would start seeing more like this outbreak getting amplified, right? And there are multiple such viruses that have emerged in the recent, I think, 60, 70 years. And I haven't put SARS-CoV-2 here, but SARS-1, Zika, MERS, Ebola, dengue, chikungunya, H1N1, HIV, West Nile. We can talk about all of those, but they are all examples of this kind. Now, the thing that I wanted to also sort of talk about is that within what happens during an outbreak, there is the data and information part of it, for which you have the vector, the host, the people who study the vectors, the people who study the host, epidemiologists, social scientists, ecologists, local authorities. Then there are people who are studying the risk factors, the ecology. Then there are people who are studying the pathogens themselves, microbiologists, molecular biologists. I largely fall into this category. And then there are obviously clinicians who are looking at the people who are affected and so on. And all of this requires disease modeling, database management, computational tools. And during an outbreak, all of these information pieces are coming together. This part of the information then flows to decision makers or policy makers who implement some kind of plan, hopefully based on this information. And then you see the effect on people. And what you hope is that some information then flows back into the community that's generating the information to get better information out. So this is what you hope are the components of an outbreak response. As you can imagine, you have to get all of this together quickly, and there are quite a lot of challenges in getting it to work seamlessly. Now, what does the kind of work that I do as a scientist is basically molecular detection of pathogens. And I use a particular tool called sequencing, which I'll tell you a little bit about, and then try and connect it back to the game that we are going to play. So if you have someone who has come to you with a fever and is not diagnosed, right? What are the kinds of tools that you have to make that diagnosis? As a clinical microbiologist, the kind of tools that you have are you look under the microscope. As you can imagine, when you look under the microscope, you need a lot of the pathogen. So the pathogen load has to be high. It has to be in the range that you can actually see it, and so on. So this is useful, but you can't do this for all pathogens. Definitely for viruses, this is not the way we look for viruses. We can grow it if you make enough copies of it, if you can grow it out of a person. But there are challenges around this as well. First, even to grow something, you need to know what it is. And if you don't know what it is hard to grow. You need to know the right conditions to grow it in. And you also, so this again is difficult, but a lot of bacteriology particularly relies on this. You can look at the host defense response. So those of you who have got dengue and got tests for dengue, one of the tests that we do is an IgM response. Are people familiar with this? So basically that's trying to look at an immune response, whether you have had a reaction to being exposed to dengue, right? And there are different types of those. So you can look at the host defense response. You can test for known organisms with molecular techniques. So for SARS-CoV-2, the test was RT-PCR. And that test is basically, we know what the organism is, we make the test for a particular organism. Now, when you don't know anything, then one of the things that you could do is this thing called metagenomics, in which we sequence everything. So you take a biological samples, you look for everything that is present in it. So how do you do this? So before that, okay, so basically before that, I must tell you what a sequence is, right? So for those of you who already know what this is, sorry for going into it, but there are some things that I want to iterate about it. So this is a structure, this is a drawing of a structure of DNA. And in that, you see that it is made up of 4 different chemicals: adenine, thymine, guanine, and cytosine. And these are modified in some ways, and they sit in this sort of helical structure. And this is what a DNA is. And it contains the instructions of all the different properties that we see. The instructions of how to get to those properties comes from the arrangement of these 4 chemicals on this string. So if you consider DNA to be a string with these 4 as 4 colored beads, what we are actually thinking of as instructions are actually the sequence of these beads. And that's why you will hear this term DNA sequence, because what it is the organization in many different patterns of these 4. There are many ways of looking at what such a string of DNA contains in your sample. This is one method in which what you're seeing is many different fluorescent dots, and these millions of dots are actually sequencing reactions going on. So even in your body, when a cell divides and it's making a copy of itself, what happens is the DNA is getting multiplied. So there are enzymes that will go sit on your DNA and make more copies of it. We do this artificially on beads, and we have fluorescently labeled molecules to understand what sequence it is coming in. The way we usually figure out the sequence is that there's a property that I didn't tell you about when I should have mentioned here. A always binds to T and G always binds to C in a double helix. What that means is, if I know that there is an A and G present in one strand, I automatically know that it's T and C on the other strand. So because using this property, there is a lot of there are technologies that help us figure out. This is called sequencing by synthesis. And each of these beads, what it's telling us is the kind of molecule, whether it's A, T, G, or C present there. And people scan these images and they figure out what's happening in each of these clusters. So this is a kind of sequencing called Illumina sequencing. This is the sequencing that I use, and I'm a bit of an evangelist for this. It's called nanopore. What happens in this kind of sequencing is that you get this pore, you get this protein molecule embedded onto a membrane, and you have DNA going through that pore. So the green molecule, the green protein is actually sitting on top, and the one at the bottom is a pore protein, okay? So it's a pore through which DNA is getting threaded through. And as it's getting threaded through, it is actually stopping the flow of ions across this membrane. And what you're getting is this electrical signal that you see on top. And from that electrical signal, we infer what chemical it is. So that's how the sequencing happens. This is actually a handheld sequencing device. It's really small, and it now comes with a really powerful computer as well. So you can take it anywhere and sequence. And okay, so people have used this to sequence in space. So that's an astronaut sequencing, I think, a tubulin gene in space. And this is a scientist sequencing in Antarctica. So that's in our lab. So that's what you see on the right there. That's the sequencer. You used to need a laptop for this. Now you don't need the laptop anymore. So we have taken this to set up a lab in Eastern UP and try to do sequencing to find out what's circulating in some of the brain infection cases there. And I'll talk more about that later, but I just wanted to tell you that using this method, what you get at the end is this kind of bunch of letters. So what you're seeing in front of you is the genomic sequence of SARS-CoV-2. So this is in a file format called FASTA, in which the first line always tells you what that thing is, and the second line, it's coming in multiple lines, but it's actually a single line of this different patterns of ATGC. Okay, and so this is what the genome looks like, and from this, if there are changes to this, that's when you see mutations, and those mutations can make a difference to how a virus behaves. So if we look at SARS-CoV-2 as an example virus, we know based on comparison to other known viruses what different parts of its genome or that pattern that I showed you code for, and so any changes to it can also be monitored by the process I showed you, which is sequencing. So for SARS-CoV-2, what you have here is the spike protein, which is in the middle. And the spike protein codes for that protein that you see on the right. That protein actually helps to bind on the cells, and the vaccines are made with that region, right? So we're particularly sensitive to any changes to that region. So if you see here, this is how mutations look like. Sometimes you get one change from A to T or G to C and so on. Can you see that dark spot right in the middle of that violet on top there? So that's the change. Sometimes you might see something like a deletion. There is a region that's missing. Mutations may or may not change the properties of the virus, because at least the properties that we are interested in. Like, for example, not every mutation is going to make changes to transmission or infectivity or fitness or lethality. Okay. And mutations in a viral genome, they accumulate over time. So every time a virus makes a copy of itself, where do these mutations come from? They come from the error-prone nature or the mistakes that are made in copying. When a virus infects a cell and is trying to make copies of itself. So because of that, every time it's making a copy of itself, there are some changes that are introduced, and these changes accumulate. So if you look at over the length of the SARS-CoV-2 genome and start looking at how many mutations, how many times does it change and so on, you start seeing that there is a lot of times that you start seeing changes across the entire genome. So mutations accumulate in the viral genome, and this can facilitate new ways of being. So what are these new ways of being? So they can be changes in transmission, they can be changes in severity of disease, they can help in vaccine escape, and so on. So at this point, I'm going to stop, and we are going to start playing a game. Is that okay? I'm happy to answer questions, but now after this, there is going to be a game which is called, how many of you have heard of this game called Plague.Inc? Has anybody heard of it? I have seeded this idea yesterday, so I know some people. Okay, good. And I'll need one volunteer to come play the game, actually, because are you okay to do that, Harish? Thanks. And what we will do is, so the premise of the game is you're a virus, and you have to come up with a strategy to kill everyone in the world. Yes. We can do a little bit. We are okay with the questions being in between the talk as well. Yeah, let's do some questions, and then yes.
So you mentioned some of the mutations would lead to change in its transmission rate. That if you're not familiar with the virus, is there some general rules?
Right. So so the way that we even know that these are the regions that code for different things, right? So if you look at that entire part, it's called ORF1AB. That just means open reading frame. It basically means not sure what's in there. Okay, so what you would do is you would take this sequence and you would match it to all other known sequence and see if anything else is known about them. For example, within ORF1AB is the enzyme that copies the virus. The RNA-dependent RNA polymerase is in that region. Yeah, so because of that, we know some properties of that region. So if there is a change in the RNA-dependent polymerase, it is possible that it changes the fitness of the virus, but we will have to do the experiments to show that. And some of these experiments have to be done in the lab. Yeah, so the error rate of a single molecule. So yeah, the question was about how nanopore is known to be error-prone sequencing, but let's ask where that error comes from. That error comes from the fact that we have to look at these patterns of electrical signals and infer what the chemical base is, and the pattern is generated as a 4 or 5 mer, depending on the pore length. Now, as you get better at pattern recognition, actually the accuracy of nanopore improves. You can take the same old data and you can take the same old data and generate better. However, at the end of the day, it is also dependent on the chemistry of the pore and so on. So I think there is a limit on how good it's going to get. So let me tell you roughly what the data looks like, right, for nanopore. For Illumina, we have a standard called quality Q30, right, the number of errors that are made, like one error in like 10,000 or whatever. If for nanopore, it's just above chance. So your chance of detection of an ATGC will be like 25%. For nanopore, it will be a little bit over that. So, but what you can do is you can stack many reads together and get a really good estimate for what's happening at that position. So with a single read, you can't get good error-free reads from nanopore. But if you have multiple sequencing reads, you get a very good estimate. So the overall accuracy of a genome sequenced by nanopore is as good as Illumina.
Gets its error, but it's actually sequencing in parallel the same.
Yeah.
Huh.
So that quality thing comes from counting how many of those simultaneous reads might be off. Can you do something like that with nanopore automatically, like sequence in parallel and infer, or is it something manual right now?
No, it's not. So maybe we should discuss this because it's slightly more technical than I had planned
okay,
here. However, we do a PCR amplification before we put things on nanopore, but the sequencing is not happening in that way, right? Yes.
How faithful is the copying? Copying happens. Is the mutation rate very small, or is it?
So I think it's 1 in 10,000. Yeah, in a single round of copying, which is why I think I came up with this number, 1 in 1 to 2 weeks. So these rates are known in the sense that you can look at a genome from one person in a transmission chain and then calculate back. So roughly, I'm tempted to say 1 in 10,000. So maybe 2 to 3 mistakes for a single genome if it's large enough. One mistake. Yeah, that's what I think. I could be off a little bit. Oh, yeah. Yes. Yeah, yes, that's correct. So for RNA viruses, it is particularly high. So it's 1 in about 10,000 roughly. For humans, eukaryotic cells, we don't allow that kind of error rate. I mean, you can't as an organism. Viruses tolerate it more. In fact, if you meddle with their RNA-dependent polymerase and make it less error-prone, their fitness declines as a population level. So there's some benefit that they are getting from exploring a larger space.
Yeah,
Yes.
I have a question.
Yes.
So you mentioned mutations accumulate in a viral genome. So just want to understand, can you elaborate on that bit? Why is it, and what is the purpose of that?
No, so basically this happens. Like if I have 2 mutations right now and then replication happens again and this virus gets selected, we are assuming that the virus is getting selected through the host. So it's not a deleterious mutation in some sense. It has got selected, able to transmit. So a lot of things have to happen. And then in the next host that is infected, you already have those 2 mutations plus the new ones that are coming up due to replication.
So is it like the mutations are selected, those mutations are selected which actually helps the virus to survive?
We don't always know. So the population of viruses that is getting selected, we only can look at the ones that are transmitting. So if there are other ones that got selected before and so on, we don't know. So at a population level, we are looking at what's going up and down. And overall, at a population level, you will see an accumulation of mutations. So this game, it's called, do people have a strong view on what their virus should be called? Yeah. Okay. So basically in this game, first you need, okay, so basically what the aim of the game is to give this virus properties that will help it just kill everyone, okay? And together we are going to decide what properties will help it do that. And first, what you need to do is choose where your disease will begin. Unfortunately, in this game in tutorial mode, you really don't have a choice. You have to start it in China. Uh, no, you'll see some of the weird things that happen. So on top of that, you will see something called news, which is about things that are happening all over the world. No, I think it was made by a person in America, as it happens, but I don't think it has a particular America-centricness in its worldview. Hard to say, but I think so. Okay, so let's start. So yes, so the plague has been unleashed. Here at the bottom here, you can see how many people are infected. So one person has been infected, and this is basically some random events that happen. You accumulate DNA points. When you accumulate enough DNA points, that's when you get to choose what properties you can give your virus. Okay, all right. So now you have 17 DNA points, and you can change the game speed to make it run faster, because if you see on top, there's a date there. So we went from 29th March to 3rd April. So yeah, run it in fast forward mode only. Yeah. So you can see all these aeroplanes and ships going everywhere, and we are accumulating DNA points. We can go to the disease menu and we can give it properties. In the beginning of the game, it tells you what to do. So you don't have choice right now, but very soon we'll have the choice. So it costs us 7 DNA points to evolve that trait, which had to do with, I think, livestock susceptibility to infection. So it increases infectivity, especially in rural regions, and mutation. Okay. No, I think right now leave it at livestock. So you'll see in China, there'll be small red spots that are appearing. So there is an increase. So there are 8 people infected. You can zoom in. So if you zoom into China now, you can see some red spots that are appearing, and you can zoom out again as well. And so things are going on. People are getting infected, but there are very small red dots that are happening. And you can look at news during the game. There'll be some news that's happening. You'll see that in the beginning of the game, the world absolutely has no clue, right? Doesn't care at all. And at some point, there'll be a switch that the world will suddenly want to make a cure. Now, if the world makes the cure faster than you can infect, then we lose the game. Okay, so do keep an eye out for what's happening. Right now, the world has no clue what's happening. But the infections have gone up to 1,000. So there are 1,500 people infected, no death. So lethality for what we are starting out with is very low. Are we playing the game now? Okay, so let's go to disease. So we have accumulated 35. So now we have the choice of, can you go to each one? So that's transmission trait, that's air. Gives pathogen ability to travel on dust particles, increases infectivity, especially in arid environments, and plain transmission. Okay, what's that water thing? Can you go to the water thing? Oh, okay, pathogen can survive outside the body in fresh warm water. Then I think there's a bloodborne transmission. That syringe is like person to person and blood type of contact. And insects, vector-borne, like rats, and I think avian. So each of them have traits. Not all of them are scientifically consistent. It's a game. But what do you think we should do? How many people feel like we should go with airborne next? Okay, 1, 2, 3, 4. Okay. How many people want to do waterborne? Waterborne. Okay, that's a roughly equal split. Okay. Okay. Anybody else wants to do blood or vector very badly right now? Okay. Waterborne people, can you put up your hand? Okay, I think this is increasing. So let's go with water. We'll do air next. Okay, waterborne. Can we do both? Do we have an option? We have enough DNA points. Okay, great. So we have done air and water. Now let's see what's happening. So you're getting more people infected. Now what has happened is it has gone from one region to another region. So you have infected a new country. Okay. So this is like infection is not going to come down. It has gone already transmitted to another place. Now, if you go into a country screen, it will tell us what that government is doing about it, what's happening, are they spending on medical research, are they distributing hand soap, what is their plan, how many people, how many healthy people do they have, how many people are infected, how many died. So this is a country specific. We can go into it in the middle of the game whenever we need to. And we can also look at the state of the world during like what's happening in the world. So once it spreads to one country, you'll see that it actually spreads to quite a few. So the world's health, this is telling us 99% of the population is healthy right now. What's happening with the cure? How far away are we from the cure? This also tells us that. And then it also tells you which countries. So if you're a strategic virus, it gives you an idea of which countries you should target in order to stop the cure. Okay. So this now we are playing the real game. Okay. So you can see one air transmission happened and lots of transmission to multiple countries happened. So already this virus is transmitting quite a bit. It's more infectious than HIV apparently. Okay. You saw that ship that went in and infected Australia. So lots of that happening. We have got enough DNA points to go in, but this virus is not killing anyone. Okay. So what do you think? Shall we change some of the symptoms of the disease? Okay. All right, let's go to symptoms. We can give respiratory symptoms, skin symptoms, some blood-related symptoms. So let's say, yeah. In the beginning, so here we again have a choice. We can make the virus transmit more by giving certain symptoms. We can also kill people, like make it more lethal. If we make it more lethal, I just want to tell you something about the game and also in real life. When people start dying, people start taking notice of it. So as soon as people start dying, you will get a cure, like people working on the cure. This just notice as it happens. So how many people for this respiratory thing? Giving it respiratory symptoms? Okay. How many people want to give it some rash? Okay, smaller number. Anybody else for any of the other things? Huh? Insomnia. Sometimes insomnia comes up by itself. So there is one insomnia particular thing that makes people less productive. I like to put it in when they are developing the cure, but for now, I think sneezing. Okay, and there's one person. Let's go with anemia as well. Diarrhea is that the stomach one? Let's go to the one on the left and give some, yeah, irritated, yeah, yeah, this will become worse. Like diarrhea in the beginning is like a very soft symptom, but later people die of diarrhea quite a lot. So, right, do we have enough points to get the next diarrhea one as well? Okay, yeah, let's do that. And what's happening is let's get some rash also, right? Some people wanted rash, so yes. Okay, what about more worse lung symptoms? Okay, we can go into that. Yeah, let's not. Let's wait. Let's go back and see what's happening in the game. Have you evolved all of these things? So now people should start dying a little bit. Hasn't happened. Yeah, check the world. We are in China right now. Nobody is dying. No, it is, we are looking at the world, no one's dying. Yeah, so we need to go back and work on the symptoms a little bit for this virus. Okay, let's make that respiratory part the one on the left. Let's see which one vulnerable. Okay, let's get that. As you keep clipping on the disease, there'll be one that people will start dying. Next, lung. So if it's a hierarchy. Yeah, go to the one on top before that. Yeah, so that heart condition. Yeah, good. Any other lethal thing that you can think of that we can do right now? What about that diarrhea endpoint and the respiratory endpoint? Yeah, let's look. If we have enough DNA points, we can make it more lethal. I think it's time. You can ask. So potentially fatal heart abnormality causes breakdown of respiratory system, releasing pathogen into the air. Potentially fatal. What is the other one? Sneezing. I don't know. What do heart? How many heart? Okay, quite a few hearts. Anybody for sneezing? Okay, sneezing people still feel we need to increase infectivity. Do we have money for both? Let's do both. Okay. Okay, let's go back. Lots of deaths, and you'll see that now this compounds really fast. Okay, now China has shut down airports. It is hard to infect people in China. Let's go to China specific and see if there are people left to infect there. Oh, if you click on China, okay, but not while, yes, get us some points as well. So when he clicks on those and bursts the bubbles, that's how we are getting the DNA points. That's why I needed someone else to do this. So as the disease is
The WHO watched this after 4 million are already dead.
yeah. They started cure, 4%. Okay, I think we have to, let's go to disease and do some demolishing of the cure. So let's go to abilities. Now this one is simulated actually as a bacteria. Okay, go to that one, the drug one. So this is, it creates a bacterial shell to protect against all climates. I don't like that one. Can we go to this drug one? Because people have started, pathogen becomes resistant to the class of antibiotics that are being developed. That's a good one. Then we have the option of pathogen becomes resistant to even more. What are the other two one and two? So pathogen becomes harder to analyze in the lab, decreases future research speed. I like that one. How many of you want to go with that one? What is the level meaning? What is the other option? It develops resistance to the second and third line of antibiotics as well. Third and fourth line of antibiotics. So that particularly affects Western countries. Okay, so antibiotic resistance versus stop research in the lab.
How does it work?
No, I mean, this is a game. We're just trying to do something to prevent the cure, right, from coming in. So I would say if we have points, select both. Actually, select all of these. It's not spreading to cold countries much, so we need to make it cold resistant as well. That snowflake thing that you see on top, that's cold resistance. Yeah, so we've made it really climate resistance now, but lethality is still poor. No, yeah, let's go back to symptoms and make it more lethal. Anybody who has thoughts, please shout out. I'll incorporate.
So this applies to one particular strain somewhere in the world, and then it's
No, it's evolving as well. So every time you are giving it a property, the assumption is it's evolving. It's sequence. And going across the world and taking it over. But it is inbuilt. You'll see that it comes out slowly. It doesn't immediately change the scenario. So I feel that in transmission, let's look at the world map. If you look at some of these arid countries, they are not getting enough infections. So I think we need to change the mode of transmission as well. Any thoughts on what we should include? Water, okay. Avian. Can we get some avian? The bird on top. Mosquito as well, I think. Yeah. Unlikely that one particular pathogen will do all of this. So this is just to give you a sense of it is hard to get something to have all of these properties in one time. So it is a balance between many different types of things. Okay. Still, I think we need to go to symptoms. The lethality is pretty low. Let's look at that heart thing in the middle. What happens? Weren't we supposed to already get, this total organ failure. How many people for total organ failure? Lots. Let's get it. What else?
kills off the most too fast, then it doesn't spread.
Yeah, exactly. So if you go for lethality before it has gone to other countries, you'll lose the game. I have done this a few times and it does happen. So this we don't know, but depends on, it may be irrelevant to the virus whether you live or not. Depends on the mode of transmission, so, and how it gets transmitted. but I don't think it would optimize for that. Most of the time, what we see is the reaction of the human body to the virus is the problem rather than the virus replication itself. That's a very broad statement. It's partially true. Okay. If countries have closed their ports and we have not infected them, we might lose the game, because how do we get there? Then we have to think of land routes, vectors, all these other things. So all kinds of transmission. I think it's time to switch on all transmission modes. Is there anybody saying anything in the chat that we should consider? No, people don't have their, okay. Somebody wanted to make it more airborne and more waterborne. So part 2 of the water, and also maybe this blood thing. This blood thing actually helps it infect lots of people close to each other. Infected bodily fluid can transmit the virus. That's what. So hospital-borne infections, accidents, those kind of things can also happen. Someone found a cure. Okay, let's, on 100 days, 34% of the population wiped out. You have to click on all those blue things as well, and we have to do something about the resistance. Yeah. So we are getting, yeah, let's make it harder for people. So the virus changed. It reassembled its genome. Yes. Yeah. Okay. So at some point, we will not be able to get lots of DNA points and make all the choices that we want. But I think we're doing pretty okay. Some arid regions are not getting, yeah, so you can see people are having trouble all over the world. Slowly governments will start to tumble. There'll be nuclear explosions. People will try to bomb cities. All of these things will happen. So we got a lot of DNA points again. Let's go and see what we need to improve on. Let's go to the world and see how the world is doing. Everyone is infected. So great. Now we just have to kill everyone off. So I think now we should work on the symptoms. Let's look what's more lethal. Suppressing immune system. Yeah, but now everyone is infected. We have to make the symptoms worse in this artificial game. Yeah, very often what happens is humans are not even the intended host. It may have an alternative life cycle and it's going on and replicating itself. And once in a while meets a human and creates problems in that person, and the person dies. Like, for example, Japanese encephalitis virus, rabies, all of these humans are dead-end hosts. They don't necessarily transmit forward. Okay, what are we evolving? If anything that says the word fatal, I think, is everyone in agreement? There are a few fatal things. Can be lethal, is better than can be lethal. Yeah, that's good. Complete breakdown of digestive system. Sorry, what was it? Immune suppression. Yeah, okay. I think that's enough. I think it's pretty much guaranteed that. Now let's look at the state of the world again. The cure is 10 years away because we stopped it quite a lot. We didn't let anyone. Who are the people making the cure? Are they infected? Everyone is infected, so we don't need to worry. Once I played this game and there was one country that was not infected and they were making the cure also, then you can lose the game. Yeah. Okay, world again. Let's see how, yeah, we are very close to the end of the game. This is the news, what's happening, okay. Insanity symptom. So yeah, now the news. is coming in about the eradication of the human race and found to be 100% lethal. So yeah, going to take longer to develop the cure. Yeah, so when the infected comes down to 0, all of them are going into death. Yeah, so this one, well done. I think all human life, not all life. Other things are evolving. So yeah, congratulations. We have won the game. So this is what happened in this particular game, right? We started with a virus that increased in severity over time. Sorry, can you go back to that disease graph? That increased in infectivity over time, that increased in severity stepwise over time, increased in lethality over time. Very unlikely scenarios in real life. Does not happen in this way, in this kind of time scale. At least, so that's what he was asking, that is it a strain that developed that and got seeded? So that depends on how realistically they model this. Ideally, at least for SARS-CoV-2, what happened was variants grew for a long time, then they suddenly exploded in populations, and then once they exploded in that population, then they went around very quickly. Because although we had stopped air travel or whatever, we were still connected quite deeply. So I think about 1 week to 10 days, you had a variant that had locally grown get everywhere quite fast. Yeah, so that is the population. And then the cure obviously didn't get there in time. So obviously this is a simulation. This is just to give you a sense of all the things that it takes. That's sort of the end of my talk. And happy to take more questions if we have time. Thank you, Harish. But it took us 535 days to do that. Huh? Like, even with our rapidly evolving mutations and.
Thanks, Chitra. So before we start the question session on, there were a couple of questions I had actually. Maybe I'll just ask you because it might generally interest people. So I remember you telling me that when COVID was on and you were trying to do this wastewater surveillance in IISc, the Ministry of Science was very reluctant to let people collect samples using this suits because they said it will start panic. So I wanted to know what kind of, non-clinical factors do you have to take into account when you do these kinds of clinical studies?
Yeah, as you can imagine, like it's very sensitive when you do go and collect samples from places, right? And like how do you appear when you're doing that sampling? For wastewater sampling, this was an issue that multiple people had, because what we were trying to do was also in like I had a company and we used to go and collect samples even from residential apartment complexes to try and understand if we were getting a signal. And if you went in with full PPE, that is PPE is personal protective equipment. So if you don the hood and the apron and gloves, etc., you're scaring people off, and more people are scared of looking at you than, and actually you're just doing sampling. And so you know that there is a certain level of protection that you need to have. You need to have gloves, you need to have a mask, and you need to have eye protection when you're collecting sewage, and you need to be vaccinated in a certain way. But if you try to do more than that and walk around like that, it was a problem. So so you have to be sensitive to what people feel and then optimize according to that. And but if you went by the book, there's often not a space in protocols for this kind of adjustment. And that's a problem everywhere, right? Whenever you bring in a restrictive mechanism, you just say no as a blanket no, but no under what conditions. We don't work with that. And in outbreaks, this is much worse. Like outbreaks, people will just prefer, if you are not sure of what you want to do, don't have a protocol in place, it's better for you not to be there. So it's very hard to try new things, or for example, take up sequencing and go, where will you fit? Those kind of issues come up quite a lot.
Which brings me to the next question. So in India, like, those of us who have worked with, public health officials often face this problem that there's a lot of denialism initially, especially like, if there's a disease going on, the first reaction would be to hide it, to suppress it. And my questions are A, like, how have you personally, like, tried to overcome this problem? And B, how does it actually prevent the eventual elimination of the disease?
So I've spoken about this to a lot of people, and we continue to speak about this. So can I do a plug in here of something that I do? So is that okay? Okay, so basically there is a podcast that we are trying to put together. It's called I Detect, and it's meant to talk to infectious disease researchers in India and people in public health space as well, to try and understand these questions better. Because it is clear that you have to work together. It is clear that you have different incentives. It is clear that the people who are detecting the disease, if they are the same people tasked with elimination or keeping that disease down, it's not going to work. So, and if you talk to any of them and work with them over time, you realize that they are aware of what's happening. So it doesn't come, so far from what I have seen, it's not some malicious intent. It is just that you know that you can't do very much. And it's really hard for us as researchers to take that. However, how do you take information and then what is the time scale at which it becomes actionable? They are two very different things. And also as researchers, we get very convinced of a certain piece of information, which for policymakers may not be information enough. They have to weigh it against everything else that is impacted. So most of it has been learning. It's also learning how to speak the language. The other part is the patience to wait and to sort of keep the faith a little bit. I mean, that's the politically, yeah, I think being a bit correct, but.
Questions?
Thank you for the talk. Personally, what I've experienced after this pandemic is that I tend to be much more conscious about keeping my hands clean and sanitation. And my parents often tell me, it's okay, it's fine. Sometimes you have to do it. But what I'm concerned about is for the new generation of kids coming up. If they are they grow up in environments which is very clean and sanitized, what would be their amounts of immunity, and would that be a concern for any future outbreaks that happen?
So I don't know about this. It's very hard to answer this, and the evidence for this is not very clear, right? Because there's so many conjectures around. We don't know if it's causal, and but I understand this idea that your immunity needs to get primed. However, let me give you a counter example that I do know about. Lots of people who get something like chickenpox, their immunity will get wiped out. So you're getting infected by something and your immunity to all previous things is wiped out. So this idea of, so I'm not sure whether being clean has anything to do with your immunity being better. Like, I don't know about that part. So I cannot answer your question, don't know. But I know that hand washing does decrease influenza. We know that for sure. So if there's influenza going around, washing your hands helps more than many other things. So I think we can only identify and answer that in pieces that we can address. Can't identify it as a whole and answer it as a whole. Does that make sense?
Thank you.
So in the context of COVID as well as respiratory diseases generally, one point that was made very heavily over the last 2 years was we need better ventilation in indoor spaces such as this. And, people talked about it, but then I think it lost steam and nobody's active. So is there any, for example, I heard IIT Bombay was seriously replanning its buildings and so on. But are people actually trying to do that?
It's very hard, as you can imagine, for people to act. There's so many other priorities that come up. It is not something of pressing, as you saw, like the cure doesn't get developed till people die. So I think that we are very reactive, and one hopes that I think there are some questions that we need to ask about ventilation, about sanitation, these kind of things, this is a good time to ask them, but this is not the pressing time to act on them. So I don't know, again, if people are acting on it, but I think we should.
This game seems interesting. How far-fetched it is, how scaremongering it is. I mean, how realistic are the game rules? Potentially, can anyone decide, maybe some AI, let such games be played on and on, content a playbook to create a perfect?
Right. So this game, I think, has been played about 130 million times or something like that. It has been around for a while now. And I think they may not have started out with such a sophisticated way. They have one that's called Kill the World. The other one is Cure the World. They have two different ways. There is a lot of information from this that tells you about how people think a virus evolves, right? Once they play a game, people, it's what the information that you're getting is not how to evolve a virus, but how people think you should evolve a virus in order to win the game. And how realistic is it? It's not too bad. The person who designed this game was consulted by the CDC. So I know that a lot of input and exchanges happened over this game, and it's not too bad. If you play the real game, we played a tutorial version, which was actually slightly lower. In the sort of next version, where if you put in a little bit of hand washing, some people being more careful, not everyone being equally susceptible. The thing is, the heterogeneity is so huge that it's hard. I would think that it's incredibly hard in real life to design something very potent without a lot of trial and error. I think that has been the experience of the world so far.
So a question that I get asked a lot often these days is, when is the next big pandemic coming? So is there any way that science can answer such questions?
I mean, I think you might be better placed to answer that question in terms of when than me. But like, let's put it this way: the risk factors and the factors that make it likely that we will see something local spread far are very high, because they are they are travel and connection based, right? And we have set the world up for that, and it's very unlikely we're going to take a backseat or should even take a backseat on that one. So soon. The idea of, at least for disease X, people are thinking that as soon as it happens, I mean, it's not when it'll happen, we don't know, but as soon as it happens, we have 100 days for response. So we had 500 days for wipeout. People are thinking about 100 days for response. So that's how people are thinking now, and to be triggered anytime. So to keep systems in sort of ready mode for that.
So like, for example, people before COVID came in, people were like looking at things like Ebola and so on as potential danger zones, but COVID essentially came out of nowhere. So how do we explain such emergence of, essentially out of blue something comes up?
Right. So firstly, I think if you ask the coronavirus researchers and people who worked on respiratory pathogens, they would tell you it didn't come out of nowhere. It was expected. Like for influenza, people monitor influenza quite a lot, like really, really avian influenza, because influenza, it's a segmented genome. And then when it infects different hosts, if two different strains infect the same host, like in pigs, I think they reassort. So you get genetic diversity really fast. And because of that, for those kind of reasons, it's a virus that is tracked a lot for its ability to maybe cause something like this. I think coronaviruses were also on that list because there were quite a few respiratory endemic coronaviruses that circulate in human populations. So I think that for people who worked on these things, you get used to the idea of constant risk. Like having worked on viral pathogens and emerging pathogens for a while, I think that it becomes hard to understand all the other complex factors at play. As a researcher, you still know very little. So, but if you went and asked them, they would, I think, say that they had expected a respiratory virus to be the problem. Nipah, for example.
So there's a lot of talk recently that because of climate change, as we have thawing of the permafrost, long extinct pathogens might be coming alive. And in fact, there's, I think, one known example of, they reviving a microorganism which was actually frozen in the tundra. So what do you think? I mean, how seriously should we take this risk?
I mean, as seriously as the risk of spillover, but then again, like that pathogen has to be competent enough in the human host as of today, and all the heterogeneity and the immune landscape that we have right now. I mean, I would say it's one more source you add to this list. I mean, I can't say anything. I can't rule it out. Let me put it that way. Unfortunately, it also depends on where the people are dying and the value of that life. But yes. Is a cure a tractable problem? I'm very optimistic about this. I actually think if you put in resources to many of these things, it's very likely you'll get answers. Whether you'll get them in reasonable time frames, but I think even in reasonable time frames, you should. Yeah, I think that technology in this field is advanced enough that we have a good shot. Well, ART actually works like quite well. Antiretroviral therapy. Yeah, but do you need, if you can live a reasonably, what it depends, right? Do we if you want to eliminate pathogens, in general, that has proven to be a bad strategy overall. But if you are willing to live with a pathogen and not get affected by too much, I think that's really possible. So it depends on what we mean by cure. And ART, antiretroviral therapy for HIV, is actually, I would think it's a success case where it has actually changed the life of someone who's infected.
Talked about some 100-day response that people have. COVID pandemic successful at getting the pharma companies to share their research with. That is not, I mean, that's caused sort of hundreds of thousands of deaths. That problem would be solved.
But what would the solution look like? Like how do you incentivize a company to share data when it's not in their interest, or there is some competing interest for them? I don't know. Then I may never form a company and I may never work on a cure. I would choose to do something else. Right? I mean, we also have to safeguard the interests of the people working. Yeah. I mean, I'm just trying to play devil's advocate here. I would like to live in a world where there is an incentive for companies to share data in a way that it benefits the maximum number of people. I think it's a consortium and that is funded by philanthropic foundations mostly. Yeah.
So slightly unrelated, but someone in the audience mentioned that it's in the interest of the virus to not be lethal, right? Spread and transmit. So is it the case that if you have multiple strains of the same virus or different viruses, they can also compete for the same host? So they would try to kill each other in.
Yeah, Omicron wiped out everything else.
So is it
wiped out as in you don't see
okay,
things anymore?
Probably,
right? Huh.
Probably not a practical, but if you have multiple strains of virus, are you more likely to get a virus naturally which is less lethal to humans?
Less lethal.
Because they would want to kill off all the other organisms which are actually killing their host, right?
Or you could what you would see is the one that has the highest transmission advantage or a huge fitness. Like we can only see, right? We don't know what's happening. Yeah. We can only see the outcome and we are making some ideas about what may have happened. Yeah. And we don't know, like we can make correlations. They may not be causal. We have to investigate it differently. So I'm not sure whether that is necessarily the case. However, it could be one of the outcomes that you see. And that would not be surprising, that if it was one of the outcomes. Again, like rabies is an example, 100% lethal in humans. There are 7 or 8 different types of rabies, very geographically clustered. Has other hosts that will transmit it. So it's irrelevant whether human beings die or not from the perspective of the virus.
Omicron gave herd immunity to most of the world against Delta and other strains.
Yeah.
But Omicron's sufficiently transmissible that it hasn't itself gone away.
Yeah. And also reinfection is possible, all these.
So, like they say that old soldiers don't die, they just fade away. So I also get often asked this question, whatever happened to Spanish flu virus? Like, so in general, what happens to viruses eventually?
Right. Is this so do you want to answer this question?
No, please.
Okay, some of them become endemic, right? So you see them at low levels. So you remember that I showed you that curve, which is background. So for example, you could get the young ones getting infected and at some level, and then they recover, then only when you have young in the population again, you have a susceptible pool again. So you could get something like that. I mean, young ones is an example, like immune naive versus someone who has an immune response, and then you will see that. So you'll always get some little bit of infection, not a lot of clinical disease, but it's there, you get a little sick or may not even notice it, you might have asymptomatic infection. I actually don't know the answer to this, but I think a related virus is definitely around. Do you know the answer to that? Is the Spanish one?
The answer is what you said, that no virus is around forever. It mutates all the time. But I mean, there are these different strains, H1N1.
Yeah, so something derived from that
basically-
virus is definitely around, but I
Yeah.
don't exactly remember.
So
this.
something very similar could arise again, possibly.
Oh yeah, I mean that.
Probably
that
not directly from the original Spanish flu, but it could arise via
Hmm.
mutations of current viruses.
I think there's a lot of interesting work going on the genetic signature of the Spanish flu virus. They're trying to exhume bodies and basically get traces of this Spanish flu and see exactly what was genetic composition.
I see. Oh, like diversity and so on.
Yeah.
Ah, interesting.
Yeah. Hopefully this is not being in China.
Hopefully it will be on live samples that are safely collected and we are only looking at the sequence and not trying to grow the virus. I mean, you know, if someone doesn't do that.
So any further questions? Oh yeah, no.
This thing that you just said, that only the or people whose immune system are not primed, but we still do get seasonal flus, right?
Immunity
Yes.
wanes. You don't get
So-
lifelong immunity.
I see. So another question that I had was, do you get pandemics only through this spillover? Like, this-
No. This is an example of zoonotic
Right.
only. How else would you get? Not from animals.
So, like, some some pathogen already present in the population, somehow it has some extremely dangerous mutation and it starts spreading.
Human, you mean human
Human,
viruses
yeah.
itself? Yeah, like for example, if Omicron picks up some.
But it's
Trait.
already a pandemic, right? By then.
Yeah, but now let's go generations after.
So, okay.
It has become largely human. Actually, that's the thing. Coronavirus is not only human. Most viruses have multiple hosts, so lots of things can happen. I'm thinking, I'll give you an example. West Nile virus wiped out the entire crow population of the US. Yeah, that's an example of something that's a pandemic in crows. Human beings, not so much right now.
Yeah, I have one more question. When it comes to policy making, the fraction of, I mean, if you look at the population of doctors, that would vary across countries, right? So how much of this would be dependent on things like facilitating communities organizing to come up with, emergency responses and, like people getting together and facilitating those kind of things? Because I've heard of these cases in certain countries. Is it like a good thing to do, or should it strictly be left to doctors and
I mean, we definitely cannot answer that in any useful way, like as in have an opinion on that. I don't have an opinion on that. However, we know what has been helpful, right? Many different kinds of, where you do not have a system set up, you need things that will do the work. Does that answer some of your question? It also depends. Are you talking about civil, like society, people
there are
coming
sometimes
together?
communities which come together, local communities, and governments tend to incentivize sometimes.
Yeah, it depends on the resources available and what you, as a policymaker, you see as implementable, right? So I would
But
say,
it's also true that experts. So
oh, that. So I don't know if you saw in this game towards the end, there will be things like a country removes all bans on animal testing, removes all bans on something else. So it depends on the risk level, right? You weigh the two risks and you make a call at that time, which may not be popular when you are put in judgment at a later point of time, but you act in a given moment with the information that you have.
But when you
Hopefully,
consult, you're mostly talking about government-informed policies.
yes,
your
yes, I would say that you need to work with people who can be held accountable and know that they can be held accountable.
How long can a virus thrive outside a living host?
Depends on which virus. So there are some viruses that, so each virus will have its own properties depending on the kind of virus that it is. Some go on for months, and some things are really bad for them, like UV might be really bad because it disrupts their DNA. But if there's enough moisture, so there are conditions under which a virus can survive longer, and depending on the kind of virus that it is. No single answer. Permafrost.
You said humans are not the intended hosts for the viruses.
Not always.
But why is it so? Like, I mean, no, why is it like humans are not the
No,
like
they may not be. As in, we cannot think of the virus as only having us as the.
Okay.
We may not be the only ones, and we may not.
Is it for like for the virus? It's a hostile kind of thing.
No. So a virus is an obligate parasite. So it requires cells to infect and grow and make copies of itself. So wherever possible, when a virus meets a susceptible cell, it will grow or try to grow in or explore the space to grow in that cell. And so if that happens to be a human cell, some things will happen. If it happens to be an animal cell, some other animal cell, it'll like, so maybe when I say, when we are talking about intentionality of virus, that's where I'm, because of trying to play a game from a virus perspective and so on. So we have to get back from that. What's happening is the chance of a virus being in a host that is conducive for its replication, which may or may not be a human.
And one more. So the gene alteration that happens in a virus, it's said mutation has a role in here. Are there any other mechanism, like, I mean, it's always random, or it's like can be also environmentally can be regulated also?
Yeah. Selection pressures, right? So in order for so suppose I'm putting in tetracycline as a growth hormone when I'm growing, when I'm domestic, like chickens or something. I want the animals to grow, and I know tetracycline is a growth hormone. So any bug that infects that animal now has to be tetracycline resistant in order to grow in that animal. So that is the environment that particular pathogen is adapted to. Like, so that's how. Does that answer your question a little bit?
Yeah, I mean, can you understand, like nowadays we are taking like different drugs and all those things.
Yeah, so it's a selection pressure.
Yeah,
that's
so that is kind of giving some stress to the virus, and that's why they are like changing to a different genome so that they can kind of.
So the stress related changes, they can happen. Usually they're documented more in eukaryotes, like higher organisms, as epigenetic mechanisms that later influence genetic mechanisms. So it does happen. However, for a virus or something like that,
I'm hinting to epigenetics.
Yeah, no, not so much. I mean, okay, not that I know of, but even as I say that, I can think of examples where this is true. So for example, in viruses, the classical example for this is bacteriophages, where there is a lytic phase and a phase that does not kill. It's lysogenic, and so integration versus, and that switch can be a stress response. So yes, there are examples of this.
Thank you.
So Chitra has other personas, like she's also a board game developer. And in fact, she's here in Chennai to participate in the board game convention that's going to take place this weekend. So as they will be showing their latest game called TESS. So she has very kindly agreed to give a preview of TESS during the high tea, which has been arranged just after this. So let's thank Chitra once more.
Thank you.
And I invite